Hi, this is Forrest Blocker. I'm going to talk to you today about cancer treatment, cancer research, what we've learned in the last 30 to 40 years, what we expected to learn, and where we're going to go from here. This is the second part in the series. When an proto-oncogene becomes an oncogene, it negatively affects your health. Proto-oncogenes normally regulate cell growth, including cell signaling. So of course cells, when they're in the environment of the tissue, they'll often have to receive signals, hormone signals or whatnot, that tells them to behave differently and make different kinds of proteins. For example, this might be adrenaline because you've had a scare because there's a bear in the room. You will release adrenaline. There will be a receptor on the outside of the cell. There'll be some transduction of this information until ultimately you will start producing the machinery to perform glycolysis, to break down glycogen into sugar so that you can have a fast response and, escape. So that's happening all the time. You have to have cell signaling. There has to be a way of communicating to the cell that it's time to do something different right now. Cell regulation, growth factor receptors, transcriptional factors, when to transcribe DNA into RNA to make some protein because you need it right now, and also anti-apoptosis proteins. Apoptosis means it's a particular kind of cell death. So sometimes when things have gone really haywire in the cell, you just want it to die. Oncogenes are constitutively active, which just means active all the time, or they're active under conditions in which the wild-type gene is not. So examples of oncogenes include MYC in small cell lung cancer and neuroblastoma, HER in breast cancer and other diseases, RAS in 25% of all cancers, SORS, ABL, BCL2, AKT, and others. Viral oncogenes are proto-oncogenes captured and mutated by retroviruses to become oncogenes. So in this picture, what we have is normally the proto-oncogene has a role to encode for growth factors. Here's a growth factor being received on the outside of the cell, just like we had adrenaline being received here. And then it's transduced, that information ultimately becomes translated into a call for a transcription to turn DNA into RNA and make some protein machinery that will do something in the cell. In cancer, the mutation caused overproduction of growth factors. The receptor may no longer be needed to turn this on, which is why it would be constitutively active. We don't need that anymore. Factors always bind to their gene target promoter, and so the stimuli is no longer needed. One kind of oncogene was called MYC. MYC is a proto-oncogene that encodes for a transcriptional factor. It can override its fail-safe mechanism. It impairs differentiation. It promotes cellular proliferation. Another one is RAS. It normally functions to promote cell growth. So here it is. We've got some receptors in a normal cell. There's some proteins that have to bind, including RAS. And then there's a transduction event, which leads to a transcriptional event. But in the cancer cell, you have this mutant RAS here. So this is the proto-oncogene, and this is the oncogene. There's no longer this negative feedback loop that we have with RAS normally. So there are three RAS genes in humans: HRAS, KRAS, and NRAS. It's the most common oncogenes in human cancer. The Philadelphia chromosome is caused when chromosome 22 and chromosome 9 become fused.
What's chromosome 22?
I don't know exactly what chromosome 22 has on it, but as you know, we have 23 chromosomes. We make duplicates of each of those chromosomes, so we have 46 duplicate chromosomes, and each chromosome has a specific set of genes on it. There are thousands and thousands of genes on each chromosome that code for different protein. Chromosome 22 is just one of those 23 chromosomes, as is chromosome 9. And they fuse so that they make this one, little protein that shouldn't normally be made. Here we see chromosome 22. That's where it gets broken. And then here on chromosome 9, it gets broken. And then the rest of this chromosome 22 and the rest of this chromosome 9 go off by themselves. But these two then come together and fuse. It's a good question. One of the first successful targeted drugs was called Gleevec. We can see here how it works.
What does it do exactly?
Okay, so this is the BCR-ABL protein that's a fusion product.
Mm-hmm.
So there's all this alpha helical part of the protein here, and there's some beta sheets up here. And right here, this is this drug right here, which is Gleevec. Okay?
Mm-hmm.
So it binds. So we can see that normally this protein binds to ATP and releases an activating signal protein, which produces leukemia. Gleevec binds right here in the active spot where ATP should bind. That means that it can't activate this signal. Does that make sense?
Yeah.
And so what we find is that with conventional chemotherapy, for people who have CML leukemia, if we start out at T equals 0 and we have 100% of all the patients, then after 1 year, not a lot of difference. After 2 years, there starts to be quite a bit of difference. After 3 years, with conventional chemotherapy, 75% of the patients are alive after 3 years. With interferon, which is an immunotherapy, it's more like 85% of patients are still alive after 3 years. But with Gleevec, it's in the high 90s. So it's a very effective drug against this disease. And it was the first really successful targeted drug that binds specifically to a targeted site. Remember that DNA gets transcribed into RNA when it's needed because of some signal that says, okay, we need to do this now. And then that gets translated into protein. And then the protein is really the machinery of the cell. The enzymes, all the working, moving parts. When a proto-oncogene, which is in DNA form, becomes mutated into an oncogene, it stays constitutively active or active during times when normally that oncogene wouldn't be turned on. So it's just continuously cranking out this protein. Okay. So the different proteins can have different functions. So there are growth factors, there are receptors for growth factors and hormones. Here'd be a growth factor, PDGF would be an example. There'd be a receptor that would recognize the growth factor, and that's also a protein that has to be transcribed and then translated into protein. Then there's intercellular transducers, like these, source, RAS, RAF. These are checkpoint proteins for cell cycle regulation. There are nuclear transcription factors,